Gene editing and genomic medicine represent one of the most transformative frontiers in human health because they address disease at its most fundamental level, the DNA code. Most diseases, whether monogenic or polygenic, are rooted in molecular errors or dysregulations that ultimately disrupt cellular function and organ systems. Technologies like CRISPR-Cas9, base editing, and prime editing allow for precise manipulation of the genome, correcting pathogenic mutations, modulating gene expression, or inserting protective genetic elements. Unlike traditional therapies which target symptoms or downstream biochemical pathways, gene editing directly intervenes at the source, offering the possibility of permanent cures for conditions that were previously untreatable. Monogenic disorders, such as sickle cell anemia, cystic fibrosis, and Duchenne muscular dystrophy, are now being approached with interventions that can restore normal protein function, reestablish cellular homeostasis, and prevent disease progression from birth or early onset. Beyond single gene disorders, polygenic and multifactorial conditions, including cardiovascular disease, type 2 diabetes, and certain forms of cancer, can be influenced through genomic modulation. While these diseases involve complex interactions among many genes and environmental factors, gene editing can potentially adjust the expression of key risk alleles, enhance protective pathways, or reduce the activity of genes that predispose individuals to metabolic dysfunction, inflammation, or tumorigenesis. This approach represents a shift from population-level interventions to precision medicine, where therapies are tailored to the unique genomic architecture of each individual, informed by sequencing, bioinformatics, and functional genomics. The precision of these interventions is complemented by advances in delivery technologies. Viral vectors, lipid nanoparticles, and emerging non-viral delivery systems allow gene editors to reach specific tissues, minimizing off-target effects and maximizing efficacy. Tissue-specific promoters and regulatory sequences further ensure that gene modifications are expressed only in desired cells, reducing systemic exposure and unintended consequences. These technological refinements make it possible to target organs that are historically challenging, such as the liver, heart, or brain, opening the door to treating neurodegenerative conditions and metabolic syndromes at their molecular origins. Ethical and safety considerations are central to genomic medicine. Germline editing, which alters DNA in reproductive cells, raises questions about long-term consequences, heritability, and unintended off-target effects, requiring rigorous oversight and validation. Somatic cell editing, which modifies non-reproductive tissues, currently presents a lower ethical risk while still offering therapeutic potential for a wide range of conditions. Preclinical studies and early clinical trials are demonstrating both the efficacy and safety of these approaches, and continuous improvements in off-target detection, epigenetic monitoring, and immune system management are increasing confidence in the long-term viability of gene-based interventions. Gene editing is not isolated. It integrates with diagnostics, data science, and AI-driven predictive modeling to identify the precise mutations and pathways that require intervention. Whole genome sequencing, combined with bioinformatic analysis, enables identification of high-risk variants before symptoms appear, allowing preemptive genomic intervention. This convergence of technologies redefines the boundaries of medicine, shifting it from reactive treatment to proactive, personalized, and potentially curative strategies. Through gene editing and genomic medicine, humanity is approaching a future where diseases are not merely managed, but addressed at their molecular root, transforming the concept of what is curable and establishing a foundation for long-term health, resilience, and human longevity. RNA-based therapies represent a paradigm shift in medicine because they allow precise modulation of gene expression without permanently altering the DNA sequence, offering a reversible yet highly targeted approach to treating disease. Messenger RNA, mRNA therapies, small interfering RNAs, siRNAs, and antisense oligonucleotides are designed to influence cellular protein production, either by encoding therapeutic proteins, silencing pathogenic transcripts, or correcting splicing errors. These molecular interventions operate at the transcriptional or translational level, providing an unprecedented ability to directly manipulate the functional output of genes, which is particularly useful for diseases caused by protein misfolding, overexpression, or viral infection. mRNA vaccines for infectious diseases, most notably SARS-CoV-2, exemplify how rapidly RNA therapies can be designed, optimized, and delivered to stimulate protective immune responses. This principle is now being extended to cancer, metabolic disorders, and neurodegenerative conditions. One of the major advantages of RNA-based therapies is their modularity and speed of development. Unlike small molecules or biologics, which require extensive protein engineering and multi-year production pipelines, RNA constructs can be synthesized rapidly based on sequence information and then delivered using lipid nanoparticles or other carrier systems. This approach allows for iterative testing, rapid scaling, and personalization. For example, in oncology, mRNA can encode neoantigens specific to a patient's tumor, training the immune system to recognize and eliminate cancer cells. Similarly, siRNAs can selectively degrade mRNA transcripts from mutant genes, reducing the production of toxic proteins that drive diseases such as Huntington's disease or certain types of hypercholesterolemia. RNA therapies also offer opportunities for precision modulation of metabolic and signaling pathways. Diseases characterized by aberrant enzyme activity or inflammatory cytokine overproduction can be targeted by silencing specific mRNAs, effectively dialing down harmful proteins while preserving healthy pathways. This contrasts with traditional pharmacology, which often lacks the specificity to affect only a single molecular target without off-target effects. By integrating RNA therapy with diagnostic genomics and proteomics, clinicians can identify patients with the exact pathogenic transcripts and design interventions that are optimized for individual molecular profiles, creating a truly personalized treatment paradigm. Delivery remains a critical challenge and an area of rapid technological advancement. Lipid nanoparticles, viral vectors, and conjugated oligonucleotides improve tissue-specific targeting, protect RNA from enzymatic degradation, and reduce immune activation that could otherwise cause inflammation or reduce efficacy. For neurological diseases, innovative delivery systems are being developed to cross the blood-brain barrier, allowing RNA therapies to reach central nervous system targets and modulate protein expression in neurons and glial cells, which has implications for Alzheimer's disease, Parkinson's disease, and other neurodegenerative disorders. RNA-based therapies integrate seamlessly with gene editing, immunotherapy, and regenerative medicine, creating synergistic approaches to disease eradication. By controlling protein production at the RNA level, these therapies provide both acute intervention for immediate pathological processes and long-term modulation of disease progression. The flexibility, specificity, and speed of RNA therapeutics position them as a cornerstone of the next generation of medicine, where molecular precision and individualized treatment convert artificial intelligence and predictive medicine are transforming the landscape of healthcare by enabling the analysis of vast, multidimensional data sets that were previously inaccessible to human cognition, revealing patterns that underpin disease risk, progression, and treatment response. Human health is governed by complex interactions among genetics, epigenetics, environmental exposures, lifestyle behaviors, and molecular signaling pathways, making precise prediction and intervention challenging using traditional clinical approaches. Machine learning algorithms and neural network models can integrate genomic sequences, proteomic profiles, metabolomic data, electronic health records, imaging, and real-time physiological signals from wearable devices to generate predictive models that anticipate disease onset, optimize therapy selection, and personalize intervention timing. By converting raw biological complexity into actionable insights, AI shifts medicine from reactive care toward proactive, preventative, and individualized strategies. Predictive analytics in medicine allows for early detection of diseases before they manifest clinically. For example, AI models trained on large-scale genomic and clinical datasets can identify individuals at high risk for cardiovascular disease, type 2 diabetes, or neurodegenerative disorders by recognizing subtle biomarker patterns invisible to conventional diagnostics. This predictive capability enables preemptive interventions such as targeted lifestyle modifications, prophylactic therapeutics, or early molecular therapies, potentially preventing the full emergence of disease and reducing long-term morbidity and mortality. In oncology, AI algorithms can detect cancer-specific mutational signatures in circulating tumor DNA, imaging, or histopathology with greater sensitivity than traditional methods, allowing treatment initiation during pre-symptomatic stages when curative outcomes are more likely. Drug discovery and therapeutic optimization are also revolutionized by AI. Machine learning can predict the structural compatibility of candidate molecules with specific protein targets, identify novel drug candidates, and optimize dosing regimens with unprecedented speed and precision. Generative models can propose new molecular structures, while reinforcement learning systems simulate patient responses, accelerating the translation from bench to bedside. AI-driven analysis of real-world evidence and post-market data further refines therapies, highlighting subpopulations that benefit most and minimizing adverse events. Real-time monitoring and adaptive interventions are enabled by wearable biosensors, continuous glucose monitors, and mobile health applications integrated with AI systems. These tools collect longitudinal physiological, behavioral, and environmental data, which machine learning algorithms use to detect deviations from baseline that may indicate early stress, metabolic dysregulation, or immune dysfunction. Personalized feedback loops inform dynamic interventions, such as adjusting nutrition, activity, sleep, or pharmacologic treatment, maintaining homeostasis, and preventing disease progression. AI also facilitates precision medicine by bridging molecular data with clinical outcomes. Integrating genomic variants with proteomic and metabolomic profiles allows prediction of individual responses to therapies, including gene editing, RNA-based treatments, and immunotherapies. This approach reduces trial and error prescribing, enhances efficacy, and minimizes toxicity, making interventions more targeted and efficient. By enabling early detection, personalized interventions, predictive drug design, and continuous health monitoring, artificial intelligence and predictive medicine fundamentally alter how we approach disease. They provide a framework for understanding human biology at a systems level, optimizing therapeutic precision, and ultimately moving toward a future where many diseases can be prevented, halted, or cured before they impact quality of life. Regenerative medicine and tissue engineering represent a transformative approach to human health because they focus on repairing, replacing, or restoring damaged tissues and organs, rather than merely managing disease symptoms. At the cellular level, most chronic and degenerative diseases are driven by loss of function, cellular senescence, and tissue degradation. Stem cells, organoids, and bioengineered tissues provide a platform to restore normal structure and function by reintroducing functional cells, stimulating endogenous repair mechanisms, and integrating with host tissues to reestablish physiological homeostasis. Pluripotent stem cells can differentiate into any cell type, allowing replacement of neurons lost in neurodegenerative diseases, pancreatic beta cells destroyed in type 1 diabetes, or cardiomyocytes damaged during myocardial infarction. Tissue loss, regenerative medicine has the potential to reverse disease progression and restore functional capacity. Organoids, miniature 3D structures grown from stem cells that mimic the architecture and functionality of organs, allow unprecedented insight into human biology. They provide platforms for disease modeling, drug testing, and therapeutic development with patient-specific precision. For example, organoids derived from a patient's own cells can be used to predict responses to chemotherapy, test RNA-based interventions, or screen small molecules for efficacy and toxicity. This technology bridges the gap between preclinical research and individualized therapy, reducing the trial and error approach historically associated with treatment development and accelerating the translation of laboratory findings into clinical interventions. Tissue engineering combines cellular biology with biomaterials to create functional constructs capable of replacing or supporting damaged tissues. Scaffold materials, including biocompatible polymers and hydrogels, provide structural support, guide cell differentiation, and integrate with host vasculature to ensure nutrient delivery and waste removal. For musculoskeletal injuries, engineered cartilage or bone can restore structural integrity, while for cardiac injuries, patches of engineered myocardium can improve contractile function and prevent heart failure progression. The combination of living cells with engineered matrices creates a synergistic effect, enhancing tissue survival, vascularization, and functional integration. Nanotechnology enhances regenerative medicine by enabling precise delivery of growth factors, genetic material, or signaling molecules directly to target cells. Nanocarriers can modulate the microenvironment, promote stem cell differentiation, and optimize tissue remodeling while minimizing off-target effects. This level of control is particularly important for neural tissue repair, where precise signaling gradients and microenvironmental cues dictate axonal growth, synaptic formation, and functional recovery. Regenerative approaches also interact with gene editing and immunomodulation. By correcting genetic defects in stem cells before implantation or engineering immune-tolerant tissues, therapies can be made safer, more effective, and tailored to individual patients. The integration of bioinformatics and AI-driven modeling enables prediction of tissue growth dynamics, vascular integration, and functional outcomes, allowing optimization of constructs before clinical application. Through stem cell therapy, organoid development, tissue engineering, and nanotechnology-assisted delivery, regenerative medicine addresses the underlying causes of tissue and organ dysfunction. These interventions restore cellular and tissue integrity, enhance organ performance, and have the potential to reverse degenerative diseases, redefining the possibilities for human health, longevity, and the future of curative medicine. Immune engineering and precision immunotherapy represent a revolutionary approach to treating diseases by harnessing and directing the body's own immune system with unparalleled specificity. The immune system is the body's primary defense against pathogens, cancerous cells, and abnormal tissue. But in many diseases, it either fails to respond adequately or becomes dysregulated, causing autoimmune conditions. By understanding and manipulating the molecular and cellular mechanisms of immune activation, regulation, and memory, scientists can design therapies that precisely target disease while minimizing collateral damage to healthy tissues. Chimeric antigen receptor T-cell therapy, or CAR-T, is a prime example, where a patient's T-cells are genetically engineered to recognize specific tumor antigens, expanding ex vivo, and reintroduced to selectively eliminate malignant cells. This approach has demonstrated remarkable efficacy in hematological cancers and is being extended to solid tumors with advances in antigen targeting and immune microenvironment modulation. Precision immunotherapy extends beyond cancer, encompassing infectious diseases, autoimmune disorders, and chronic inflammatory conditions. Monoclonal antibodies and bispecific antibodies can be engineered to recognize specific proteins or cells involved in disease pathogenesis, modulating immune responses with high specificity. For autoimmune diseases, immune checkpoint modulation and regulatory T-cell expansion strategies are employed to restore tolerance while suppressing pathological inflammation. Vaccines designed using mRNA or peptide-based platforms provide another layer of immune engineering, allowing rapid adaptation to emerging pathogens and personalized targeting of antigens based on individual HLA types and immune repertoires. These interventions work at the molecular level, influencing cytokine signaling, antigen presentation, and lymphocyte activation to achieve therapeutic precision. Engineering the immune system requires integration with advanced delivery technologies and computational modeling. Nanoparticles, lipid carriers, and viral vectors allow controlled delivery of immunomodulatory agents directly to lymphoid tissues, tumors, or sites of inflammation, improving efficacy while reducing systemic side effects. Machine learning models analyze patient-specific genomic, proteomic, and immunologic data to predict optimal targets, dosing, and combinations of therapies. This predictive capability ensures that immune interventions are both personalized and adaptive, dynamically adjusting to changes in disease state or immune responsiveness. Immune engineering also interacts synergistically with regenerative medicine and gene editing. Genetically modified stem cells or organoids can be designed to evade immune rejection or to secrete immunomodulatory molecules, enhancing graft survival and tissue integration. Similarly, gene editing of immune cells can enhance their cytotoxicity, persistence, or specificity, expanding therapeutic potential across a range of diseases. The combination of immune modulation, cellular therapy, and precision targeting creates a multi-layered strategy for addressing conditions that were previously untreatable. The ultimate goal of immune engineering and precision immunotherapy is not only to eliminate disease, but to reprogram the immune system for long-term resilience. By establishing durable immune memory, correcting dysregulated pathways, and selectively enhancing protective responses, these approaches redefine therapeutic possibilities, offering a future where infections, cancer, autoimmune conditions, and chronic inflammatory diseases can be precisely controlled or potentially eradicated. Neurotechnology and bioelectronic health interfaces represent a paradigm shift in medicine because they provide the ability to monitor, modulate, and optimize neural and physiological circuits in real time, creating opportunities to treat diseases that were previously resistant to conventional therapies. The nervous system is central to nearly every organ system, regulating cardiovascular function, metabolism, immune responses, and hormonal balance, as well as controlling mood, cognition, and motor activity. By interfacing with neural circuits through invasive or non-invasive technologies, such as brain-computer interfaces, transcranial magnetic stimulation, deep brain stimulation, and wearable bioelectronic devices, it is possible to restore function, recalibrate dysregulated systems, and provide continuous feedback for precision interventions. Brain-computer interfaces, BCIs, translate neural activity into actionable signals, allowing individuals with spinal cord injury, stroke, or neurodegenerative disease to regain motor control, communicate, or interact with their environment. Advances in electrode design, signal decoding, and machine learning algorithms have enabled higher resolution and specificity, allowing precise targeting of motor, sensory, and cognitive circuits. BCIs not only restore lost function, but can also be used diagnostically, mapping neural activity patterns associated with disease progression, neuroplasticity, or cognitive decline, thereby informing treatment strategies and rehabilitation approaches. Non-invasive neurostimulation methods, including transcranial direct current stimulation and transcranial magnetic stimulation, modulate cortical excitability and plasticity to treat depression, chronic pain, epilepsy, and cognitive impairment. By adjusting the frequency, intensity, and location of stimulation, these technologies can selectively enhance or suppress neural circuits, effectively recalibrating dysfunctional networks. Combined with neuroimaging and electrophysiological monitoring, these interventions allow dynamic tuning of neural activity, optimizing therapeutic outcomes while minimizing side effects. Wearable bioelectronic devices extend this capability beyond the brain, targeting peripheral nerves, cardiac tissue, and metabolic organs to regulate physiological function. Vagus nerve stimulation, for instance, can modulate autonomic tone, reduce inflammation, and influence gut-brain signaling, impacting conditions such as heart failure, rheumatoid arthritis, and depression. Closed-loop systems integrate real-time physiological measurements, such as heart rate variability, glucose levels, or neural activity, with automated stimulation protocols, creating adaptive feedback loops that maintain homeostasis and prevent disease exacerbation. Neurotechnology also integrates with regenerative medicine, gene therapy, and immune engineering. Neural prosthetics can guide stem cell integration, enhance synaptic connectivity, or deliver localized molecular therapies. Bioelectronic interfaces can modulate immune responses by targeting neural immune circuits, offering novel approaches to chronic inflammation and autoimmunity. Machine learning algorithms analyze the vast data generated by these devices, predicting disease trajectories, optimizing intervention parameters, and personalizing therapy to each patient's unique physiology and neural architecture. By enabling precise monitoring and modulation of neural and physiological circuits, neurotechnology and bioelectronic interfaces provide a mechanism to restore function, optimize health, and prevent disease progression across neurological, metabolic, cardiovascular, and immune systems. These approaches represent a convergence of neuroscience, engineering, and computational biology, forming a foundational pillar in the future of curative, personalized, and adaptive medicine. By integrating these 6 pillars, gene editing and genomic medicine, RNA-based therapies, artificial intelligence and predictive medicine, regenerative medicine and tissue engineering, immune engineering and precision immunotherapy, and neurotechnology with bioelectronic health interfaces, we move toward a future where diseases are addressed at their root causes rather than merely managed. Each of these strategies interacts synergistically, enhancing cellular function, neural circuitry, immune precision, and systemic resilience, while leveraging technology to deliver personalized, adaptive interventions. The convergence of biology, engineering, and data-driven medicine creates unprecedented opportunities to not only cure previously untreatable conditions, but also prevent disease, optimize human function, and extend health span, demonstrating that the future of medicine will be proactive, precise, and transformational for humanity.